All right, welcome to another week of Soul Seminar Online. Today I'm introducing Michael Dzakovich. He's at the USDA-ARS Children's Nutrient Center, Research Center, Department of Pediatrics at Baylor College and Medicine at Houston, Texas. Michael's research interest lies at the interface of plant genetics, analytical chemistry, and human nutrients. His overall goal is to leverage environmental manipulation and natural variation in these plants to determine which chemical properties of food crops affect human health while defining underlying genetics that control these traits. This goal will be accomplished by combining genetic and analytic techniques, targeting and untargeting omics approaches, and in vitro models of conceptualizing findings. Through these approaches, Michael seeks to illuminate poorly understood aspects of plant metabolism, optimizing growing conditions of phytochemicals of interest, and provide readers with genetic targets that can be used to develop nutrients-enhancing crops. And when you're ready, you can go ahead and share your screen, Michael.
Okay, thanks so much, Chelsea.
Yeah.
Okay, am I on?
Okay. Looks good.
Okay. All right. Yeah. Thank you so much, guys. Really appreciate the invitation to speak today. So today I'm going to be talking about some of the work that I actually did as a PhD student with Dr. Jessica Cooperstone at The Ohio State University. And I think part of it is because Matt Gibson had reached out to me and asked me to talk about some of the work that I did using metabolomics. And so I think this is a really cool story that you guys are going to enjoy, and has maybe a little bit of a different twist because I'm going to be talking about an animal study that I did as well. So before I jump into everything, I wanted to kind of briefly introduce myself because I know of you from Twitter and whatnot, but haven't had the pleasure of meeting in person. So I wanted to talk a little bit about my journey here. So I started off working with tomatoes back at Purdue University, where I did my BS and MS working in a plant physiology lab that was run by Carrie Mitchell. And I was mostly looking at how different wavelengths of light affect the nutritional and sensory properties of tomatoes. I then moved on to The Ohio State University, where I worked with both David Francis and Jessica Cooperstone, looking at how genetic diversity influences the chemical properties of tomatoes. I did a brief stint at NC State University at their Plants for Human Health Institute. Unfortunately, not working with tomatoes, I was working with sorghum, but it was still an interesting time looking at how we absorb phytochemicals in the gut and how we can change the genetics of plants to change that process and make it a little bit more efficient. And then now I'm working for the USDA-ARS, but I also have a joint appointment with the Baylor College of Medicine. So it's a little bit confusing. I have two bosses, twice as much paperwork. To make matters worse, I recently discovered a clerical issue where my job of plant physiologist was changed to plant psychologist. So if any of you have any plants that are undergoing biotic or abiotic stress, my door is always open to unpack those traumas. Okay, so enough about that. Now that you guys have kind of a brief understanding of who I am, I wanted to kind of start big picture of why I did all this work in tomatoes in the first place. So globally, they're an extremely popular fruit or vegetable, depending on your perspective. We consume, as Americans, we consume close to 40 pounds of them a year, most of which are in a processed form. And because of that, they have a very high value across the world. And I've been using this image on the right for a long time, but I think with Thanksgiving coming up, it's finally relevant. So not only are tomatoes economically valuable, but they're also a host to an array of different phytochemicals. And probably the most famous phytochemical is lycopene, which is the red colored pigment in tomato fruits. The interest in lycopene started way back in the mid 90s from a very large epidemiological study that was done. And it involved about 48,000 healthcare professionals. And so this was a prospective cohort study, which in the spectra of epidemiological studies is a very powerful study design. And essentially what they were doing was looking at how dietary patterns influenced health outcomes in the long run. And what they ended up finding were that multiple tomato products and tomato containing things were associated with a decreased risk in developing prostate cancer. And so what they kind of took this reductionist approach and they said, okay, what's something in common other than that these contain tomatoes? And what they came up with was lycopene. And most of the lycopene that we consume as Americans, at least, I think about 80% of it comes from tomatoes. So they kind of figured, okay, lycopene is sort of unique to tomatoes. It's in other things too, like watermelons, but it mostly comes from tomatoes in our diet. So maybe that's the thing that's having this impact. And so this spurred a ton of research over the past few decades, looking at the effects of lycopene and health outcomes. So one of the first studies that I wanted to show you here it's from almost 20 years ago. It was done at The Ohio State University. And so this figure that you're seeing is a survival curve of rats that were destined to develop prostate cancer. And so what they did was they had different diet treatments. So there's a control group, a group that received a diet supplemented with just lycopene, and then a group that was supplemented with tomato powder. And what you can see is that lycopene had an effect. The survival was a little bit higher towards the end of the study, but whole tomato had a bit of an edge. And so that kind of tells us, okay, maybe there's just not enough lycopene, or there's something else in tomatoes that's having this effect. And so a logical next question that was studied a little bit later by Dr. Jessica Cooperstone and others was what happens when you have more lycopene coming from the diet. And so if you're familiar with tangerine tomatoes, these are an orange colored, naturally occurring tomato that produce cis isomers of lycopene that are absorbed almost 10 times better than the trans lycopene that's present in red tomatoes. And so using a tomato like tangerine, you can test this hypothesis to see, okay, what happens when you have more lycopene coming from the diet or circulating or depositing in tissues that you want to study. And so in this study, mice were exposed to ultraviolet light to develop skin cancer. And over time, the tumor number and size were tracked. What you can see here is there's 3 different diet groups. You have mice that were on a control diet, mice that were on a diet plus tangerine tomato powder, and mice that were on a diet plus red tomato powder. And what you can see here is that by the end of the study, there isn't a significant difference between the 2 tomato groups. But when you compare tomato to the control, you can see a 50% reduction in tumor number. And again, I just want to back up here. This is from adding tomatoes into the diet. This isn't adding a medication or a pharmaceutical. This is adding a vegetable. So this is a pretty big finding because it kind of tells us, okay, it doesn't seem like more lycopene is better necessarily, but there's still something about tomatoes that's having an effect. And so this kind of leads us to the next question of what other compounds are playing a role. And so again, Matt had asked me to talk a little bit about metabolomics today, and I think this is really a great example of where metabolomics can play a helpful role in answering these questions. So this leads me to the big question that's going to kind of spur the first part of my talk, which is what compounds from tomato are absorbed and deposited in the liver. And I mentioned the liver specifically because this is the primary metabolizing organ in mammals. This is one of the first stops where components from our diet are absorbed, accumulated, metabolized, and then either stored some more or distributed to the rest of the body. So it's really a great tissue to study in terms of looking at disease outcomes in the long run. And again, a great opportunity to use metabolomics to study this. So I'm going to kind of give you a brief overview of what metabolomics is. I'm borrowing this slide and adapting it from Dr. Jessica Cooperstone because I think it really illustrates this point so well. And so we're all kind of familiar with these first 3 ohms, and there are other omics as well. If you can pretty much put any word in front of ohm these days, and probably someone has studied it. And of course, all of these are kind of interacting with each other and changing each other. But the metabolome specifically is kind of the chemical fingerprint of a sample or a tissue or an organism at a certain point in time. And so the metabolome can include a lot of different types of compounds like amino acids, lipids, carbohydrates, hormones, nucleic acids, primary and secondary metabolites. And it's also highly influenced by both your genetics as well as the environment you're in. And because of that, your metabolome is always on the move. And so for me, if you took a blood sample of me at different points in this talk, you'd probably see chemical changes over time just from me going through this presentation. Okay, so we've given you kind of a brief overview of why we're interested in tomatoes and how metabolomics can help us answer questions about the health benefits of tomatoes. And so I wanted to start off by talking about the animal study that I alluded to. This was a collaboration that really went quite literally across the campus at Ohio State University. There are people on here that are in the horticulture department, the food science department, and even the medical focusing on oncology. So it's really kind of embodied this whole crops to clinic approach that again helped us answer questions, kind of fundamental questions about the health benefits of tomatoes. So people kind of asked this question before of what happens after you consume a tomato, but there's still a lot of unanswered questions in the literature. So particularly when you look at other papers, people will often use animals that have mutations throughout the genome that can affect other metabolic processes. So it makes it a little bit difficult to apply some of these outcomes to what actually happens with people. From a gene expression standpoint, most of these studies use very targeted approaches like qPCR. So you can only really see the changes in expression of genes that you expect to see. And from a chemical profiling standpoint, most people use very targeted approaches. So they're looking at compounds that they know are going to change, like carotenoids, but you might be missing out on some of the other things that are happening in the background. And so we wanted to take a little bit of a different approach. So in this study, we used wild type mice. So this will hopefully make our findings a little bit more broadly applicable to other organisms and to people. For looking at gene expression, we use RNA-Seq to get a more global picture of what's happening with the transcriptome. And for our chemical profiling, we use untargeted metabolomics. And again, that's just to allow us to see a more a holistic view of the chemical fingerprint of these liver samples in response to their diet. Okay, and so to kind of go over the experimental design, we had 3 different groups of mice that were fed either a control diet, a diet supplemented with tangerine tomato powder, or a diet supplemented with red tomato powder. And after 10 weeks, they were sacrificed and their organs were divvied up. And specifically in my case, I used the liver for both RNA-Seq analysis as well as untargeted metabolomics. So I'll start with some of the untargeted data first. So I'll be focusing on a data set that was collected from a methanolic extract. And so this is more polar and semi-polar compounds. And then the ionization mode that I'll be presenting is ESI positive. And so this is, you can either have positive or negative, and usually you see more compounds in positive. I used a software called Agilent's ProFinder to deconvolute our spectra, which is kind of a fancy way of saying find peaks and chemical features within our data. And then everything was analyzed in R. So I wanted to walk you through kind of a typical workflow that's used in untargeted metabolomics, because unlike some of the other omics that I mentioned earlier, we don't necessarily have like a reference genome or a reference metabolome to compare things to. So we have to kind of go from scratch every time. So in this study, we started off with 9,041 chemical features. That doesn't necessarily mean we have 9,000 chemicals. It could be adducts and other things that can occur within a mass spec, but we need to kind of bring that number down so that's something that we can reasonably work with. So after doing a pre-processing step, which helps get rid of features that might not have been in all of our QC samples or had very high variance, we get down to about 2,500 features. And so again, this gets us a little bit closer to that one-to-one relationship between a chemical feature and a metabolite, because we've kind of gotten rid of some of the other ones that might be, for better or worse, noise. So from there, we used a battery of different statistical tests, such as some univariate tests, as well as multivariate statistics, to kind of come up with a unified list of chemical features that are highly influencing our data sets that we can prioritize for identification. So the first thing I wanted to show you here is a principal components analysis, and this is a scores plot. And one of the first things I wanted to point out is that you can see these blue triangles here. These are our quality control samples. And so in an untargeted metabolomics study, it's really important to see that your QC samples are clustering together because it means you didn't have a lot of within batch variation. The next thing I wanted to point out is that you can see a pretty big separation between our control samples, which didn't receive tomatoes, and our treatment samples, which either received diet supplement with red tomatoes or diet supplement with tangerine tomatoes. And furthermore, you don't really see a lot of separation between the two groups of samples that consume tomatoes. And so for us, we did a number of univariate statistics. And so this was really good evidence for us that we could kind of pool these two groups together and just kind of compare control versus tomato fed. And so of those univariate statistics, one of the first ones I wanted to show you was this is a volcano plot from t-tests that we did. And so to orient you, on the x-axis, you have fold change of how much a chemical feature changed relative. And then on the y-axis, you have a p-value. And so essentially what you can see is that most of the chemical features that we saw that are significantly different are on the right side here, which means they increased. And so for us, that kind of tells us these are probably derived from tomatoes themselves. The other thing too is that if you actually look at these p-values here, they're really, really big. And part of that is because we saw a lot of present-absent relationship. And so again, that kind of helps drive home that hypothesis that these are actually coming from tomatoes and being accumulated in the liver. And so again, kind of going back to that initial question that I had of what compounds accumulate in the liver is a response of eating tomatoes. This figure here really helps drive that point home. And so this is a heat map that was constructed with chemical features that had a high VIP score or greater than 1. These are considered to be truly significant for a number of multivariate statistics. And what you can see down here is that there's this kind of large swath of chemical features that are almost totally absent in the controls, but present in either of the tomato-fed groups. And so again, going back to that original question, this seems like a really good place to start for compound identification because we're clearly seeing a presence-absence relationship with these chemical features. And so I'm summarizing quite a lot of work here, but essentially what I ended up doing was going through using databases to kind of predict chemical formulas and also using MSMS experiments to look at the fragmentation patterns of these individual peaks. But what I found within that little cluster that we saw in the heat map were 61 individual features that belong to steroidal alkaloid metabolites. And so most of these were identified using this MSMS approach, where we basically went through and blew these molecules apart and looked at the individual pieces of each of these peaks to try to understand where it's coming from. We were able to identify tomatidine and dihydrotomatidine by authentic standards because those are available, but the vast majority of these don't have authentic standards. And so they're at what we consider as a level 2 identification, which means that we have an accurate mass match, but we also have some fragmentation data as well to back that up. And if you're not familiar with phase 1 and phase 2 metabolism, this is a process that happens in humans and other mammals as well, that happens both in the small intestines, but also in the liver. And it's essentially a way for our body to kind of detoxify itself. And so when I say detoxify, it doesn't necessarily mean that the compound it's acting on is poisonous for us. This happens to all sorts of other phytochemicals like flavonoids or phenolics, and even some vitamins that are important for our health. So in phase 1, these are pretty simple reactions that are done by cytochrome P450s that basically involve moving hydrogens or hydroxyls around. Phase 2 is a little bit more complicated, and these involve actually tacking on chemical moieties to compounds to make them more water soluble so that we excrete them in the urine. Okay, so I covered the untargeted metabolomics portion of that talk, and I wanted to touch on the RNA-Seq data as well. So for our RNA-Seq, we used 100 base pair paired end reads at a depth of 40 to 50 million reads per sample, and I used EdgeR for differential expression analysis. And so I wanted to show kind of a quick workflow of what we did for RNA-Seq. And you can kind of, if you remember back to the untargeted metabolomics workflow, that was all kind of done from scratch. But in this case, we actually have a reference genome that we can align our reads to so that we can actually figure out what gene a certain transcript belongs to. And then from there, we had a number of comparisons that we made, comparing our different treatment groups to each other, as well as our combined tomato versus control. Okay, and so kind of like when I showed the PCA figure for our untargeted data, I also wanted to look at kind of the big picture of what's happening with our RNA-Seq data. And so I use multidimensional scaling here. And what you can kind of see that's different about this versus the other one is that there really isn't a lot of separation that we're seeing between our different treatment groups. And when I first saw this, I was kind of a little bit concerned. I was kind of disappointed. But then I started thinking about it and I realized, well, we're asking kind of a subtle biological question here. We're basically asking what happens when you eat a vegetable. So we wouldn't really expect the entire genome to change in response to eating a tomato or any vegetable. So in a lot of ways, this is kind of a good sign that we're not seeing a ton of change in response to our treatments. So kind of hammering that point home, I have a couple of Venn diagrams here that show both the upregulated genes and the downregulated genes in our different treatment group comparisons. And as a whole, only kind of a small percentage of the genome changed in response to tomato feeding. And again, probably a good thing, but it's still a few hundred genes, which is enough to be confusing in terms of trying to figure out what's going on. And so because of that, I ended up using something called gene set enrichment analysis, which helps kind of group these different genes that are differentially expressed into biological processes. And what you can see here is that these were all of the processes that were affected. And depending on what treatment group comparison you looked at, you'd have maybe a slightly different group for the genes that were differentially expressed. But one of them was common throughout all of them, which is this drug metabolism by cytochrome B450. And if you remember, I mentioned that phase 1 metabolism is conducted through these enzymes. So again, the genes that we're seeing, the categories of biological processes that we're seeing in relation to our differential expression is all related to phase 1 and phase 2 metabolism. So these two data sets fit really nicely together. Okay, to kind of summarize here, we found 161 chemical features that differentiated between tomato and non-tomato fed mice. Steroidal alkaloids comprise the most distinguishing features between mice-fed control diets and mice-fed tomato diets. And 19 of these masses were phase 1 and phase 2 metabolites of steroidal alkaloids that were identified at levels 1 and 2. And again, most of these metabolites haven't been reported before, which is really exciting. And lastly, tomato consumption modestly affected gene expression, but the differences that we did see were all related to phase 1 and phase 2 metabolism. So again, these two data sets fit really, really nicely together. Okay, so I'm coming back to this slide kind of as a checkpoint because we've talked about what happens when you eat a tomato, at least from the lens of a mouse. And what we ended up finding were all these metabolites of steroidal alkaloids. And so again, that kind of is a bit of a recurring theme because this wasn't the very first time that steroidal alkaloid metabolites have been found. Other studies, such as the one that I mentioned by Dr. Cooperstone, have also found some of these metabolites before in animal tissues. And so this really, at this point, we're going to think, okay, maybe this is important. Maybe this is worth looking at. So I wanted to introduce what these compounds are to give you guys some context of some of the next experiments I'm going to talk about. So steroidal alkaloids are cholesterol-derived metabolites, and particularly there's a suite of them that are unique to tomatoes. What I'm showing you here, I've highlighted one of them. This is alpha-tomatine. This is probably the most well-studied steroidal alkaloid, although I would say in general, these aren't a particularly well-studied class of metabolites. I've also highlighted two parts of the pathway here that correspond to what I'm kind of considering early pathway steroidal alkaloids and then late pathway steroidal alkaloids. And so this figure from Cardenas et al. Really nicely shows that as the tomato ripens, goes from green to red, you'll see a shift in the profile of these compounds. And so what do these actually do in the plant? So they're phytoalexins. They're also able to help the plant defend themselves against pathogens. Some of the earliest work that goes back all the way to the 1940s. So people have been looking at these for a little bit while, but they really haven't received a lot of attention until the last several years. Some folks have done cell culture studies with these using different cancer cell models and have found that these compounds are able to reduce the growth of various types of cancers. And I had written in one of the previous slides that it wasn't thought that these compounds were absorbed before, partly because they can bind to cholesterol. And it was thought that they were always excreted after being consumed in the diet. And then additionally, tomatine, which is the aglycone of alpha-tomatine, so just this steroidal backbone here, has been shown to reduce muscle atrophy in both in vitro and in vivo models. So what don't we know about steroidal alkaloids yet? And so again, we're kind of interested in these from a nutritional standpoint, but some of the basics haven't been covered yet. And one of those is how to actually extract and quantify these in tomatoes. Secondly, we don't have a really good understanding of the chemical diversity of these compounds. And we don't really have a holistic understanding of the genetic architecture that controls these compounds. And so again, if we're interested in these from a nutritional standpoint, we need to be able to actually breed tomatoes to have different levels of these. And so all of these three questions kind of with each other and need to be addressed. And the first one that I want to talk about here is how to actually extract and quantify them. And so this was a method paper that came out in June 2020, and I collaborated with a PhD student in Dr. Cooperstone's lab, Jordan Hartman, who did a lot of great work on this project as well. And so what are some of the current issues with quantifying these compounds? There really aren't any validated, until this paper came out, there really aren't any validated methods to do this. And it was actually very slow to extract them because you had to do everything kind of one at a time. From an analysis standpoint, there really weren't a lot of sensitive methods to detect these. And moreover, a lot of them weren't very selective because you'd end up having to hydrolyze all these compounds and basically pool them all together as tomatine essentially, and kind of use that as a surrogate for measurement. And so my goal was to come up with a high throughput extraction and analysis method that we could then use to quantify these compounds in tomatoes and tomato products. And so here I'm showing you a chromatogram from my analysis method. And so each one of these peaks corresponds to a different steroidal alkaloid. And I mentioned when I was talking about the mouse livers that there were really only two standards available for these compounds, which makes method development difficult. Normally, what you would do is you go out and you buy a standard, you run it by itself, you'd be able to study it at different voltages, different temperatures, different collision energies. But in my case, I kind of had to work backwards and kind of make this kind of quality control sample that was comprised of over 30 different types of tomatoes, and then see how many of these I could find, and then optimize their parameters afterwards. And so one of the things that really helped me develop this method is the fact that steroidal alkaloids fragment in predictable ways. And so what you're seeing here is MSMS spectra of alpha-tomatine. And so again, this is that bigger compound that I had highlighted in that figure earlier. And below is it's a spectra from an actual sample. And so what we can do is we can use these spectra to compare against each other. And we can say, okay, given the fact that this is at virtually the same retention time and has the same MSMS spectra, we can conclude that this is the correct compound. And some of the other steroidal alkaloids, they might not have the exact same mass, but some of them produce some of the same fragments. So we can at least say, we know what these are based on their parent mass, and we know that they're making some of these characteristic fragments. And so that helps us identify what they are. Okay, so now that we had that analysis method, we could go through and develop that extraction method that had a really high efficiency. What you can see here is that for alpha-tomatine and alpha-solanine, which is a kind of the potato version of alpha-tomatine, we had really high extraction efficiencies, which is great. We also determined the limit of detection and the limit of quantification. And just to stress how low this is right here, this is 1 femtomole within our injection. So you can have a very low level of this compound in your sample and still be able to quantify it with, or at least in this case, detect it using our method. One of the next things that we measured was how much this method varies day to day. And I won't go through all of these, but essentially all of these numbers are relatively low and considered to be a really robust method. And so again, you can use this high throughput method to very quickly extract and profile these compounds in tomatoes. I mentioned that Jordan Hartman had helped me with this project, and he did something that I thought was a really, really fantastic addition to some of the kind of more classical method development work. And so what he did was he went to a number of grocery stores and he bought many different types of tomato-containing products. And we used our method to quantify steroidal alkaloids inside these products. And essentially what we ended up finding was that on a per serving basis, you're getting close to 0.7 to 3.4 milligrams per serving, which is pretty appreciable for a phytochemical. And so these compounds are actually, as far as tomatoes go, they're everywhere and anywhere. Okay, so to kind of summarize this experiment, we developed and validated a high throughput extraction and analysis method, and we generated some of the first reports of steroidal alkaloids in tomato-based products. Okay, and so I mentioned that we had kind of these 3 overarching questions about what's not known about steroidal alkaloids. And so we've accomplished how to extract and quantify them, but now we can use that method to better understand the chemical diversity and the genetic architecture that's driving these compounds. So this brings me to the last experiment that I want to talk about, which is looking just at that, trying to define the genetic architecture and understand the chemical diversity of these compounds in the red fruited clade. And so essentially what we wanted to do was put together a diversity panel of tomatoes to better understand how genetics and the environment interact to generate diversity within this crop for steroidal alkaloids. And so what we did was to assemble this diversity panel. I utilized data from a previous study, Blanca et al., that assessed over 1,000 different types of tomatoes that were really from all over the world, but mostly from Central and South America. And they were genotyped by a 7,720 SNP array. And so as part of the genetic analysis that was done on that initial diversity panel, they did something called a rarefaction analysis. And to orient you, you have a number of individuals on the x-axis and number of alleles per loci on the y-axis. And so this is kind of a surrogate for genetic diversity. And so what you can kind of see is that after a certain point, you can keep adding individuals within these groups, but you don't necessarily get more genetic diversity. And so we utilize these data to kind of help us estimate how large our subpopulation sizes needed to be in order to capture the most amount of genetic diversity. We also utilized some neighbor network analysis that was done that classified these different tomatoes into different subgroups. So again, we could sample from these different subgroups to try to maximize genetic diversity. And so what we ended up with was a diversity panel of 107 accessions of tomato. These included 25 accessions of Solanum pimpnellifolium, which are these wild small fruited species that you can see here. We had 32 accessions of Solanum lycopersicum variceraciforme, which is an admixture between modern day tomatoes and pimpnellifoliums. Then we had 51 different accessions of both fresh market and processing tomatoes. And these were grown out in 3 different environments with 2 blocks per environment for a total sample size of 642. And again, I just wanted to kind of show you guys the geographic distribution of some of these different tomatoes that we had used for this diversity panel. And these are specifically the wild tomatoes that were in it. And so kind of to give you guys a little bit of a context for what this study looked like, here's a picture of one of our field environments. So again, we had 3 of these grown over a couple of years. And you can see here in this nice illustration that we really had a lot of diversity, both in shape, but also in size of tomato fruits. Okay, and the first thing I wanted to show you was just kind of a general sense of the diversity of these compounds in our different groups of tomatoes that were in this diversity panel. And so this is this box and whisker plot is total steroidal alkaloids. And one thing I want to alert you to is that the y-axis is actually on a log scale. So the diversity of these compounds is actually really, really massive, like multiple orders of magnitude. But we did that to make interpretation a little bit easier. But generally speaking, what you can see here is that some of our cultivated varieties of tomatoes tended to have lower amounts of these as compared to some of the wild tomato species that we were working with. And again, I mentioned that our long-term goal is to actually breed for these compounds. And so from a breeder's perspective, heritability is an extremely important metric to know. And so we use this equation here that was developed by Cotterill back in 1987 to determine the genetic contribution to these traits that we're measuring. And what you can see here is that for most of these compounds, the heritability is extremely high, which means that it's relatively easy to breed for these. Okay, and I wanted to show you guys a principal components analysis loadings plot of our diversity panel. And what you can see here, I wanted to point out first, is that you have all of these blue triangles that are clustered together, and these correspond to our cultivated processing varieties. And again, we saw that they had, relatively speaking, the lowest amounts of them, but also it appears the least amount of diversity as well. Another thing I wanted to point out about this PCA is that you can kind of clearly see 2 groups forming here. There's, most of your wild species are kind of in this cluster here, but you can also see that there's about 10 groups of tomatoes that are off kind of doing their own thing. And so that tells us that there might be something happening within our diversity panel that's driving this separation. So on the right here, I'm showing you a loadings plot, which is essentially showing the contribution of the different variables that went into this principal components analysis. And what you can see here are some of these late pathway steroidal alkaloids. And on the left here, you can see some of these early pathway steroidal alkaloids. And so again, this kind of follows the shape that we're seeing in the scores plot, where we're seeing this kind of separation between 2 of our groups of tomatoes. And so here's kind of a rendering of the steroidal alkaloid pathway. And I wanted to highlight here again, here's the early part of the pathway and the late part of the pathway. And again, that corresponds to what we were seeing in that loadings plot, showing that separation between these 2 parts of the pathway. So I also did a number of correlation analyses between all of the analytes that we measured. And one of the first things that I wanted to mention is that all of the nearby analytes, so kind of neighbors within the pathway, are all highly correlated with one another, which makes sense. But we also saw some weak negative correlations between some of the late pathway intermediates and some of the early pathway intermediates. But the last figure that I showed was actually the whole population. And when we look at just the wild cherry tomatoes, which were the tomatoes that we saw kind of separating off on their own, we can see much stronger negative correlations between some of these late and early pathway intermediates. So again, this kind of tells us that there's something about these wild cherry tomatoes that might be driving this difference. And so just to kind of hammer that home a little bit more, I wanted to show you just kind of some box and whisker plots of an early pathway intermediate. So you can see here in this left cluster, we have a much higher amount than on the right. But when we look at one of the last intermediates in the pathway, we can see that that trend reverses. So again, there seems to be coordinate regulation happening within this pathway. So the next step is that we performed a GWAS on this diversity panel, and we found 302 putative associations between SNPs in our GWAS and the traits that we were measuring. And so again, from a breeding perspective, that gives us a lot of genetic targets that we can potentially use to modulate the levels of these compounds within the fruits. But doing a GWAS by itself, there's a lot happening. There's a lot of markers that were significant. Some of those could be false positives. We don't know. And so what we did was we wanted to create a validation population. And so we did this by taking a cultivated processing variety, 8243, and we crossed it with LA2213, which is a high alpha-tomatine accumulating red fruited tomato. And we analyzed both the F1 and then generated a backcross population from it as well. What you can see here is that this histogram of alpha-tomatine distribution within the population, you can see that there's a number of individuals that retained that high alpha-tomatine phenotype in the ripe fruits. And again, seeing high alpha-tomatine in ripe fruits is kind of unusual because it's supposed to, as ripening progresses, it's supposed to go down and be converted into those late pathway intermediates. And so this tells us that these individuals might have some kind of mutation or something going on that's preventing them from converting alpha-tomatine to those late pathway intermediates. One thing I also wanted to mention too is that in some of our initial crosses, we also wanted to just get some kind of basic genetic information. And what we found is that low alpha-tomatine in fruits is a dominant trait. And so in these box and whisker plots that I'm showing here, on the x-axis, we have our different parents in our cross. And on the y-axis, we have concentration of alpha-tomatine. And what you can see here is that in this case, we had two different parents. We had Tainan, which is a cherry, and 8243, which is a processing variety. What you can see is in both cases, the F1 hybrid had essentially the same amount of alpha-tomatine as the low alpha-tomatine parent. So again, this tells us that this trait is dominant. And so this is actually, I apologize for the title goof up here. So this is actually a QTL mapping analysis that was done on the backcross population. And so we used a different marker set. This is actually a much smaller marker set that's been curated to have very polymorphic markers specifically for cultivated processing tomatoes. And we use this marker set to validate that backcross population to see if we were seeing the same QTL pop up as we were in the GWAS. And indeed, what we ended up seeing was markers at the end of chromosome 3 that were associated with some of these early pathway metabolites like dihydrotomatine, alpha-tomatine, hydroxytomatine almost, and acetoxytomatine just kind of peaking over that significance threshold. But what we also saw are a number of SNPs that were associated with some of these late pathway intermediates. And again, this kind of goes back to that idea that maybe there's coordinate regulation in this pathway happening. And so again, coming back to that schematic of the diagram of the pathway, what we're seeing is that QTL on chromosome 3 tended to be associated with this early portion of the pathway, whereas QTL on chromosomes 10 and 11 were associated with these later parts of the pathway. Okay, to summarize this experiment, we determined that steroidal alkaloids are very diverse within different groups of the red fruited tomato clade. And we also created populations of tomatoes that are high in alpha-tomatine that we can use for various breeding goals, such as testing nutritional hypotheses about these compounds. We also found that tomato steroidal alkaloids are under strong genetic control, which again, from a breeder's perspective, is very important because it means that it's very viable for us to actually breed for them. We also determined that QTL on chromosomes 3 were associated with some of these early pathway metabolites, and QTL on chromosomes 10 and 11 were associated with some of these later pathway metabolites. So to wrap up this whole talk here again, Matt had kind of asked me to talk about metabolomics, and I technically did that for the beginning portion of the talk, but I'm hoping that you can kind of see the value of using untargeted metabolomics to spur questions that are related to plant metabolism or plant nutrition. And so again, what we went over today was we determined how tomato consumption affects biological processes in the liver using a mouse model. I talked about a novel methodology to extract and quantify steroidal alkaloids, which were compounds that were found to be significantly differentiating our different treatments from that mouse system. And then we utilized that method that I developed to better understand how diversity affects the profiles of tomatoes, specifically in the lens of steroidal alkaloids. And with that, I have a lot of people to thank for these different projects, specifically Dr. Jessica Cooperstone and her lab group, as well as Dr. David Francis and his lab group. And a special shout out to Sean Fenstermaker, who helped me when I was working on that crossing block. Together we literally made hundreds of crosses in a very busy semester. So I really appreciate Sean's help with that. I'd also like to acknowledge the Clinton lab, who provided the material that was used in the mouse study, as well as the North Central Agricultural Research Station, which was where we grew all of our field tomatoes, the Department of Food Science and Technology, MCIC South for bioinformatics support, and of course, all of the friends that I roped into helping me pick tomatoes for multiple summers. And lastly, our sourcing, the Department of Agriculture, for some of the funding that was used to provide my PhD program, the OARDC at Ohio State, and the Foods for Health Discovery team at the Ohio State University. And before I break there, I figured I'm a new enough PI. I feel like I can kind of do this shameless, quick self-promotion here. I'm just starting up my own lab, as Chelsea mentioned. Probably going to come up with a more creative name at some point, but something more descriptive. But I guess I'm the only member right now, so I guess that is pretty descriptive. But what I'm gearing up to do is phytochemical profiling, and also using some of the skills that I developed as a postdoc, using in vitro digestion models to better understand how we absorb some of those phytochemicals that are of interest. And I also have access to a really cool controlled environment facility, which is a 2,200 square foot facility on the top of this building here. You can see we have our greenhouse up there. It's 2,200 square feet of greenhouse and pretty fancy growth chambers. So I'm looking for collaborators always. And if you found this talk interesting and want to work together, please send me an email. So with that, I'd like to wrap up, and thank you all for your time.
Awesome. Thank you so much, Michael. So if you have questions, you can just type question in the chat box, or you can type that out if you'd like me to read it. Stacey already has a question, so if you want to unmute your mic.
Michael, thank you for that. That was absolutely beautiful. Like, it just, like the amount of work in there, and it's just fascinating. I loved all of it. So I'm going to have to rewatch I cover a lot of these similar themes in my class. And so we had read the Butelli paper about the tomatoes and the mouse lifespan. And it's neat to see kind of that there's lots of other things in tomatoes that are responsible for the effect. We also talk about like entourage effects. So this is a perfect thing to put in my class. Okay, so I guess I don't have so many questions. Two, one is as far as breeding potential, I thought these things were all pretty bitter, and I wonder how much we can actually sneak into people's foods without them thinking the tomatoes taste bad. And my second question, and after I'll mute myself, is what are these wild cherries? What's their provenance? What's their genetic background? Why are they just these crazy different metabolite diverse group? So those are my two questions.
Yeah. So I'll answer your questions in reverse order. So the wild cherry species that I alluded to, and as well as others that weren't even part of that project, they mostly come from South and Central America. And so this is kind of the point of origin of the tomato, right? And so that's where you can kind of find the most amount of genetic diversity, because these aren't necessarily domesticated yet. And so they haven't gone through all these breeding bottlenecks that give us some of the, highly productive and delicious tomatoes that we have today. And so that's partly why you see some of this diversity in chemical metabolism within these tomatoes, because they just have some of that original diversity still there. And then I guess for your first question, so that's a really good point about the taste. So I guess maybe one thing is that I don't know if we know for sure if these are the cause of bitterness. They're certainly associated. know that 100% for sure. Charles Rick did report that there were some high alpha-tomatine tomatoes that weren't bitter tasting. So that's strange. So that could be maybe a bitter blocker or something. We don't really know that yet. I think someone is working hard on that. And then I guess secondarily is tomatoes. We kind of have the luxury with tomatoes in that very amenable to being processed into a variety of products. And so even if these compounds were bitter tasting, I'm sure food scientists could find a way to make a pretty tasty beverage that would be able to mask or cover that flavor so that it could be used without any issues in a clinical setting.
Thank you. That was super, super interesting.
Cool. Is that the hint? Is Jessica Cooper stone? Is she working on the bitterness? Is that what's going on? Cool. Maybe we can invite her later.
Yeah, that's true. It's another graduate student in my group is working on this now, and so I can give like a little teaser because I don't think it's a secret. But what we're doing is basically continuing to create these backcross populations such that we have isogenic material like with and without alpha-tomatine, so that we can really do the right sensory test you would need to do to be able to say bitter or not, and then use that sort of as we develop this kind of material for testing nutritional hypotheses about what alkaloids might be doing in terms of a health context.
Awesome. Super cool. All right, Federico, you have a question? Unmute yourself.
Yes, hi, great talk. I have two questions. The first one is related to the domestication and the SGAs. So the story is that during domestication of tomato, potato, there wasn't a reduction of SGAs in the fruits and in the tubers because they are toxic. So, but as you said, these substances are important for the plants to defend themselves against pests and things. So do we know what happened with the vegetative tissues? Did they also lose the SGAs, or they still have it in the same levels? Is more a regulatory thing, or?
Right. That's a really good question, because I would imagine that there are probably, I mean, this is the same for carotenoids even. There are, fruit specific for different genes in that pathway. So I would imagine that it's probably a similar case here where, if people were selecting either purposely or inadvertently for, kind of this chemical profile in the fruits, it's hard to say if that would translate over to the vegetative tissues as well. I mean, I think we know that, I don't know from a diversity standpoint what this would look like, but I know for sure that there are a lot of these compounds in the leaves and in the roots and in the stems. So they're certainly still there, but yeah, relative, comparing like cultivated leaves to non-cultivated leaves, I've never done that before. I'm sure someone has, but yeah, that's a good point.
Okay, and the other question is about your QTLs in chromosome 3. I know that there is an SGA cluster in there. The genes are clustered there in chromosome 3. Is it the same region? Or?
So yeah, that's a good question. So yeah, I think actually, I think that was a talk a few weeks ago. So I think you're alluding to that locus that was found in chromosome 3. So we don't know for sure if that's the exact same locus that we found, partly because the marker set that we use ended before that locus begins. So where we were seeing significant markers was right at the end of our set of markers on chromosome 3. So it could have been picking up that signal, but we generate additional populations and enrich markers in chromosome 3 to confirm that. But yeah, at this point, we're not positive, but it is quite likely that is the same signal, because some of the parents that we used were, I think, some of the same ones that were used in that study as well.
Okay, thank you so much.
Awesome. Any other questions? Pablo, you can unmute yourself.
Thank you. Hi, Michael. Thank you for your talk. That was really amazing. I have two questions, I think. One of those is related with these populations you described, the the wild populations of tomato, and it's about this locus in the chromosome tree and the transporters that are associated to the SGAs in the cells. Do you think the difference you found in these populations could be related to that? That maybe, yes.
Yeah, so I think, yeah, this is kind of similar to the last question where that locus that was discovered on chromosome 3 recently, I think that they had determined that it was a transporter that allowed, I think, yeah, okay, so backing up, I think what they've determined is that this pathway is in different parts of the cell. So part of it occurs within the vacuole, and then part of it occurs within the cytoplasm. And so I think the idea was that as ripening progresses, alpha-tomatine gets shuttled out of the vacuole and then can be acted upon by those enzymes that are in the cytosol. And so, yeah, it could be. I think prior to that paper coming out, we were thinking that maybe the locus that we were seeing was maybe related to like an ethylene responsive transcription factor, because that, that kind of seems, ripening related. So, it could have been something like that. But yeah, the transporter hypothesis is really interesting. And that's something that, yeah, again, we haven't confirmed that for sure. But that is something that we're, working towards.
Okay, and you think there could be some kind of ecological association to find that associated to these populations that have some differences in the chemical compounds in the SGAs. Do you think there is some kind of ecological effect that drives that these populations are so different?
Yeah, that's a really good question. So yeah, one thing I want to mention is that all of these tomatoes, so there were 10 tomatoes in that kind of separate cluster that we found. They were all from almost the same place. And so it kind of, it's not like one was from Mexico and one was from Colombia, and it was like they were all from the same like region of Peru. So that kind of tells me, okay, something must have happened at some point in that area, and for some reason that was, those alleles were kept. I don't know what ecological consequence it might have. One of the things that's interesting is that these bitter tomatoes, they're still out there. They're basically land races that local people still grow to this day. And they, I've tried them. They are pretty funny tasting. But what's interesting is that people in that region that grow these tomatoes, they've been eating them for eons. And there's no, as far as people can tell, there's no like consequence to that. There's no like, incidence of people getting some like weird disease or something like that. So that's part of the why, even though the kind of analogous versions of these compounds in potatoes, for example, are really toxic to us, but for some reason, the slight chemical differences in these compounds, they might not act on the same pathways in people. And so there really isn't a lot of evidence that they're toxic for us. So that's something that, certainly, continuing my work with Jess from my PhD, and, kind of, we don't know that these are what's curing diseases when you eat tomatoes, but, it kind of seems like there really isn't a toxic effect from these. So maybe there is something to it, or maybe something beneficial.
Okay, thank you.
Is there any other talks? Some people had to leave, that's fine. People really enjoyed your talk, Michael. I did too. It was great. It was super comprehensive, and we're all impressed with all the work that you've done to get there. I think that's it for everyone. So thank you again, Michael, for presenting. Just a note for our UK, I think other places in Europe, I think this Sunday you have a time change at the end of your daylight saving time. So just a reminder, the seminars will actually be an hour earlier than it is this week. So be paying attention to your emails. Next week, Elizabeth McCarthy will be presenting on the evolution of flower color in Nicotiana alkaloides from pigments to genes to modeling. All right, have a good weekend, everyone.
All right, take care, guys. Yeah, thanks for the invitation. I appreciate it.
Thanks.
All right, see y'all.